Hi, good afternoon, everyone. Good morning, everyone. And behalf of Shiva family, I would like to say thank you so much for giving us a wonderful opportunity to explain our analytical services expertise, especially for the nitrosamine, followed by the E&L as a part of EPA and drug analytical and drug product the services. So today's meeting agenda is the company overview followed by general setup for E&L studies, the inhalation and the nasal product, vice versa case studies, then nitrosamine assessment for API and drug product, and last but not the least with the question and answer session. So while going forward, I would like to request you if you have any concern, doubts or questions, kindly ask in the Q&A session or else you can put into the chat box you have in the Teams. So accordingly, we can discuss after our session all those questions answer. And I believe after this session, you will get fruitful expertise and knowledge about the E&L followed by the nitrosamine for the API and drug products. So Dana, next slide, please. So attendees, today's speaker to completed this session with me, my colleague, Mr. Suman Datta is there, who is taking caring the Pharma Lab managerial activity, followed by Dr. Kiran is there, who is the operation head for Pharma Lab and the QA point of view. So about the Cotecna, I would like to share some important points with you. Cotecna, we are a leading provider of a testing, inspection, and certification services dedicated to the making supply chains safer and more efficient. We are headquartered in Geneva, Switzerland, with the offices and lab worldwide. We have over the 6,200 employee services in 180 offices and the laboratories across the overseas. Our expertise spans across five key sectors, namely food and pharma, agriculture, metals and minerals, certifications, and ESG solution and consumer products. I would like to addition to this, I want to say that we are celebrating our 50th year anniversary. Follow us the social media for insight into our journey and key milestone for the typically testing and inspection and certifications industry. So I am requesting my colleague Mr. Satya kindly explain about the Shiva Analytical as a part of Cotecna in India. So now floor is yours. Satya, you can go ahead.
Thank you, Dr. Navish. Good afternoon and good morning to all the wonderful audience here. Thank you so much for your presence, and I would like to brief about Shiva Analyticals. Shiva Analyticals is basically a contract testing laboratory which is located in Bangalore, Karnataka, which is also IT capital of India. We have been existing in this location for almost past 3 decades, and we've been supporting the manufacturers in different business verticals like food, environment, materials testing, ocean mills exploration, in addition to the pharmaceutical services. Our pharmaceutical laboratory was inspected thrice in our history by US FDA based on the data submitted by our customers. So next slide, please. We have global footprints spread across the globe with all the customer base from these locations. We are receiving samples from these customers based out of all over the world. The next slide, please. So with this outset, I'd like to address capabilities and competence and nitrosamine evaluations, which is termed as cohort of concern by IC. So basically, the nitrosamines are required to be evaluated in all the drug products. This extends to extractable leachable solutions also. Since we would like to discuss more about the nitrosamine piece as a part of E&L evaluations. So with this small introduction, I would like to leave the floor to Mr. Suman, who is an E&L expert, who will take forward and discussing the extractable leachables in detail. So Mr. Suman, floor is yours.
Good afternoon and good morning, all. It's a great opportunity to be present over here. And thank you, Shiva Analyticals and Cotecna, for giving us this opportunity so that we can share whatever we are doing over here and what are the flow and all those things. Those are for the extractable study for any regulatory submissions and all those things. So I'll initiate with the extractable and leachable study. So initially, the extractable leachable study, which is a general study scheme overview. Initially, for any query for extractable leachable comes, we must first assess the material for what we can get as much knowledge as from the vendor or the supplier of that material. then we can use, if the informations are not available, we can use GCMS with the TDU to get the informations about the material and that additives, all those things. Then you can design an extractable study by calculating the analytical evaluation threshold, which is based on the SCT as provided by USP and PQRI guidelines. And we define analytical techniques such as GCMS, LCMS, ICPMS, IC for different analytes like VOCs, non-volatiles, semi-volatiles, anionic impurities, and elemental impurities also. Followed by, we can go for leachable studies or simulation studies, which will be done under using placebo or formulations under conditions of accelerated stability testing. We have to come to know that leachables are generally a subset of extractable, but it is not always like that. Some other components might come into the drug formulation which are not extractables. So we have to finally we have to give the relevant extractables and leachables, and whichever we have to correlate, we have to correlate and report those things. So there's for any extractable leachable, the recommended guidelines or regulations we can speak of are PQRI guidelines, CDR guidelines from FDA, EMA, USP 1663, 1664, ISO 10993. So these are some guidelines for which deals with extractable and leachable testing, and also BPOG guidelines for single-use systems. So this is a typical example. In order to initiate the extractable study, we have performed direct analysis of plastic material using TDS GCMS. TDS is nothing but thermal desorption system, where we have taken two plastic material. One was printed and one was not printed. This help us to identify critical leachables prior to extraction studies. So from our study, we found that in the printed material, we got some of the compounds like cyclohexanol, butyl glycolate, which helps us in distinguishing between a good material and a poor material. Later, for the extractables, and we can correlate whether it is leaching into the drug substance or not. So this other part of that packaging material was a rubber stopper. There are two rubber stoppers. So in the initial one, we found one degraded product of butylated hydroxytoluene, which was 1,3-dietert-butyl benzene, and in comparison to which is the material used for the preparation of the rubber stoppers. And also for the other, we found some components like benzyl alcohol and naphthalene. And as we know that any PNA or PAH are highly carcinogenic in nature, so it helps us to distinguish that this material is of poor quality and cannot be used for the packaging materials, because this carcinogenic substance might leach into a drug formulation and rendering its efficacy and all those things. So once we get an idea about what the container closure system is made up of, we can design an extractable study depending upon various solvents and various extraction techniques. So the solvents we can choose depending with the based on different polarities, non-polar and mid-polar or polar solvents with buffers with different pH, acidic pH or basic pH. And the extraction techniques can be used like oxalate extraction, reflux, incubation, autoclaving. But the main thing we have to keep in mind that our substance should not decompose in that solvent. That entity of the substance should remain intact. So there are some, with this, after the extractions, we can use several instruments like HSGCMS for VOs, volatile organic compounds, ICPMS for elemental impurities, LCMS for non-volatiles, GCMS derivatized or non-derivatized for semi-volatiles, and others. So these are some quantitatively how we can approach for any extractable and leachable study, that we have standards which are known to known, can be quantified for the known to known standards. And if the standards are not available, with the internal standard, we can quantify those individual extractables. Or we have a set of medium response factors. We'll have a set of standards. We'll calculate the response factors, and for the unknowns, we can calculate based on those RRF calculations. Another thing is that for any standards, we can inject the standard at a AAT level. If you see that the blue line, that is the AAT level, and anything beyond that line with the response, we have to report as extractable coming above 8. So these are the three main approaches for the analytical ENL study for quantification of extractables. So techniques, as I already discussed, these are typical some techniques used for the extractable leachable studies like TDS, GCMS, MS. which is used for screening of VOCs, oligomers, additive decomposition products, and other unknowns. Phase-based GCMS is mainly used for residual solvents and volatile or in aqueous extracts. GCMS is used for qualitative and quantitative analysis of extracts and derivatized extracts, specific analysis like oligomers, additive, including their breakdown products. For LCMS-MS, we analysis for extract like semi-polar, polar compounds, spinning of polar known possible, but there's not always any perfect tool to analyze all those things. So we have used all the techniques for identification of extractables that comes after the container closed systems. ICPM is mainly used for the elemental impurities, other dyes and pigments. Some other techniques can be used for extractable study also like NMR, FTIR, HRMS for structure elucidation of any unknown leachables or extractables coming into picture. So this is a typical example, as I was telling, that if you see from this diagram that GCMS-MS is mainly used for non-polar and polar derivatives. ICP-MS is for ionic impurities, whereas LCMS-MS with ESI or APCI mode, we can covers a wide range of the components from non-polar, mid-polar to ionic instruments, and hence we can conclude that not a single technique cannot be utilized for extractable leachable study. We have to utilize all techniques available with us to get a proper idea, clearer picture of what are the extractables or leachables coming from the container closure system or is there in our drug formulation. So this is one of the things, the AET principle. We know as per the PQRI guidelines, they are given two constants, the safety constant threshold or the SCT, which is 1.5 mg per day for PODPs, the parenteral and ophthalmic drug products, and 150 nanogram per day for YNDPs, that is orally inhaled and nasal drug products. So if you go through the chart for PQRI or USP, you can see that there are some risk-based chart is there, the highest depending on the interaction of the material with the human consumption. So this is an application scenario where the ophthalmic product was maximum daily dose was 0.5 ml, and for nasal drug product it was 2 ampoules, which was equal to 4 ml per day. Now the AT came out for the ophthalmic product was around 3 mg per ml, whereas for inhalation it was 375 nanogram per ml. So we can see that for inhalation products the AT is much lower and compared to the ophthalmic products, and the risk is also very high for any orally inhaled or nasal products compared to other parenteral or ophthalmic products. So this was one of the extractable for a polyethylene component we have found, and mainly this homologous series of polyethylene oligomer. It gave us some minor compounds of some VOCs, some typical additives, and some additive degraded products like Irigafos 168, Iriganox 1010 is degraded product, BHT, and Iriganox 245. So these are the antioxidant which was mainly used in the preparation of this polyethylene material. So we got that. So, and as I was telling the simulation study can be performed with the placebo or drug product formulation under conditions of accelerated stability samples. Example, storing for 1 month at 40 degrees centigrade and 75% RH. And then we can follow all the techniques, different techniques we have been discussing like LCMS, HPLC, GCMS, ICPMS, and all those things. And the sample patient and all those things use what are already mentioned previously. So here is the case study for inhalation products that we found in our over here in our in-house. So it was a nasal drug product which was mainly aqueous formulation. The experiments were Avicel RC591, which was used as dispersant, buffer glycerin, benzalkonium chloride, which was used as preservative, polysorbate 80 as a non-ionic surfactant. The maximum daily dose for the product was 400 mg or 0.4 ml. The container closure system mainly consists of a polyethylene bottle of 20 ml, and other components were there like cap, stem gaskets, housing, chapel, silicone, springs, deep tubes, and water stems, and all those things. So the threshold, as we have seen earlier, that threshold for nasal drug products or inhalation drug products is 150 nanogram per day as per PQRI. The AT came out to be 375 nanogram per day. Now, for determination of worst-case extractable profiles of respective material by CCS was used with the TDS GCMS-MS or thermal desorption GCMS. The control extraction procedure was done with the three simulating solvents like IPA water in the ratio 1:3, and pure water, type 1 water at 50 degrees centigrade for 72 hours. The screening of the final extracts was done with H-based GCMS, GCMS for semi-volatiles, and LCMS for non-volatile extract. The same thing was compared with placebo with a simulator study, which was kept for 1 month and 40 degrees centigrade. And we found that similar components have been leaching from the drug product, from the contact closed system into the drug product. So the main source of extractable to the bottle was a deep tube. IPA was one of the extractable, which was formed due to the hydrolysis of triisopropyl borate, which was used as a catalyst, polysiloxanes from silicon gaskets, and processing additives from bottles and deep tube are the predominant extractables that were observed in this study. Nitrosamines and APS and drugs. So nitrosamines is part of the extractable leachable study also. So it mainly came into picture in queries and all those things in 2018 when some of the drug products, mostly the certain drugs, were recalled from the market due to its presence. And we also know that as per ICHM 7, it is a COC or covert of concern chemical and it is highly mutagenic and carcinogenic in nature. In nitrosamines can be from not only during synthesis of the drug products and also from degradation of any API, like for example, ranitidine, it degrades to NDMA, and also from packaging materials like nitrocellulose laminated blisters. So the main things are the mutagenic impurity risk assessment we have to do for the nitrosamines, whether its formation is possible or not, and all those things. The need of nitrosamine monitoring in drug substance and drug products, and we have to develop a high sensitive method and also validate those method depending upon requirement of the regulatory. And also for my side, I'll just show you the nitrosamines in relation to the ENL assessments. So this is a typical example how a nitrosamine is formed. Like any nitrate ion will be there, which will be hydrolyzed to nitrous acid, which further hydrolyzes to nitrosyl cation. And in presence of any secondary alkyl amine or aryl amine, in this nitrosyl cation will convert to in nitrosamines. So some of the amine sources are mostly the secondary amines are highly risky for this case, along with that the tertiary amines which can get degraded to secondary amines, for example, triethylamine, diisopropyl ethylamine, aromatic amines, catalysts, solvents, impurities. Those are there for formation of nitrosamines, and also some of the solvents which are used in the preparation of API or some other forms like DMA, PNMP, quaternary ammonium salts, and other additives, which leads to the formation of nitrosamines. So for packaging materials, in case of rubber stoppers, mostly used for meter dose inhalers, we know that those rubber stoppers are curated, and mostly the accelerators they are using like dimethyl sodium disulfide or 2-mark-benzothioazoles and all those things. On heating of this type of compounds, they convert to secondary amines, which further leads to formation of nitrosamines in presence of any nitrosating agents. So here are the few of the examples of the old or new accelerators or vulcanizers that are used in rubber stoppers. And the red marks most probably the sites at which it breaks down to form the secondary or tertiary amines which get degraded to secondary amines and results in the formation of nitrosamines in presence of any nitrosating agents like HMT, DPG, NS. So here are a few more examples of the accelerators or vulcanizers from where nitrosamines can be formed. We know that in rubber accelerators, a lot of tertiary amines which can easily degrade to secondary amines are used during the curing of rubber. So this might lead into the formation of nitrosamines also. So from nitrocellulose blister foils, which is used as in packaging, mainly many pigments, mostly azure compounds, which are used for nitrocellulose blister foil. This contains nitrogen, and the structure may contain secondary or tertiary amine as a functional group. So some UV curing agents like Irgacure 369, Irgacure 379, and Irgacure 907, from where we can get a secondary or tertiary amines which can degrade to secondary amines and thus becomes a potential source for the formation of nitrosamines in the drug product. And so the risk assessment for nitrocellulose packaging are there. So if it is present, then we have to further go whether it is coming from the print or whether it is coming from on the other sources, that all adhesive or anything that we have to go. And if nothing is there, then no risk is required for assessing. So as we know, as per USP 1664, it is already mentioned that nitrosamines and PNS, pH, and 2-marketobenzothiazole, that there is no limit for them, and we can detect as much, and we should detect as much lower as possible. For the determination of nitrosamines, we'll rather consider LOD rather than AET of any product. Like for example, for LOD, you have considered DCMS as 30 mg per liter, and we should determine whether any secondary amines is present or not, or any tertiary amine which can degrade into secondary amine are present or not below that LOD level. If anything is coming above that LOD level, we should take proper actions for that. So presence of so we have the main concern for us is that we should identify all the relevant and secondary tertiary amines, any materials, or whether any container closure systems we are using, and we should report all the secondary and tertiary amines above LOD rather than above AET. When secondary amines are present, and further we should investigate the material for any presence of any formation of any N-nitrosamines with targeted and sensitive analytical method. There are two options are there, whether we can choose a set of nitrosamines and we can develop a method and validate that method and confirm that, or we can go for a broader detection of nitrosamines compounds. So if no amines are detected more than LOD in the extractable study, then there is no immediate concerns, and presence of nitrosamines from the packaging perspective, no direct consequences for leachable study is there. But it doesn't mean that nitrosamines cannot be present in the drug product, because as we have seen, like for example, ranitidine, that drug product itself degrades to forms in nitrosamines. So we have to check that also. So if any nitrosamines are directly detected in material extract, then we should first step should we should change that material of construction for that. And if no material change is there, then we should monitor that it doesn't leach into the drug products. So there is always a general concern about the presence of nitrosamines in the drug products. One of the potential sources of drug products in packaging of the drug products, as we already mentioned, and historical cases are there, like for the rubber stoppers and other elastomers used for the MDIs and other packaging materials. We have found that formation of nitrosamines are possible for them. And if we have any idea about the packaging material previously, whether any secondary or tertiary amines are present in that packaging, it will be helpful for us to assess the same. And we should always focus on the precursor of nitrosamines and the formation of that nitrosamines above LOD for secondary and tertiary amines, and potentially monitor secondary and tertiary amines during leachable studies also. So with this, I'll summarize for the extractable leachable study. We should consider all the parts for the contact closure system, especially during the initiation of study. We should consider the primary packaging materials, and if anything migrates later on during the stability batches, we should consider whether it is coming from the primary packaging material, or then we have to go for the secondary and tertiary packaging materials also for testing. And we should align all the extraction and screening methods too, like what are the material types involved and all those things. The AT for sufficient signal sensitivity, that our instruments and all those things would be highly sensitive to go as much lower as possible. And we should use our worst case extraction, realistic worst case extraction conditions to be applied. Uh, we should be real so that whatever the extractables coming out of the material, that should be correlated with the leachables. And additionally, simulation study or leachable study considering the new and unexpected leachables like migration and secondary leachables that I already told that we should go later for other testing also. Sometimes the matrix interferences from the placebo and the for drug formulation is there. So our analytical methods should be developed in such a way to avoid those interferences, and the black selection should be such that no interference are there in the blank itself, which might give a false positive or false negative results. And as per because of the regulatory submission is required for extractable leachable study, we should always maintain our GMP for monitoring the stability and variety target methods ideally. With this, I'll conclude my part, and to take you through the nitrosamines in a deeper way, I'll ask Dr. Kiran to continue with that. Thank you.
Thank you, Suman. Hi, good afternoon and good morning to everybody. This is an great opportunity for me to explore our capability for testing of nitrosamine impurities at our facility on behalf of Shiva as a part of Kotakna. Uh, as our colleague Suman explained about the basics and the synthesis and all the things about the nitrosamines, so now I am going to take that much time for basics. And in my session, I'm majorly going to cover the daily, uh, day-to-day challenges where when we are analyzing the nitrosamine impurities. Okay, as a my case studies. Before that, if you look into the discovery of nitrosamines were first discovered in the year of 1950 as the contaminants in the food products. Nitron in the 1970 regulatory has banned as a food additives in many countries due to their carcinogenic properties. Okay. Subsequently, studies have found nitrosamines in other consumer products, leading into the concern and regulatory action. Okay. After that, understanding the role and risk of nitrosamine in drug safety. These are the some things we need to mainly focus for the drug safety. Those are nothing but nitrosamine in medications. It includes nitrosamines are organic compounds present in the medications, human diet, and environment, which raises the concern for patient safety. Also, carcinogenic classification. These compounds are classified as probable carcinogens, necessitating the stringent safety methods in pharmaceuticals. Pharmaceutical safety challenges, also chemical overview for the nitrosamine impurities and historical context and detections. Next. Come to the analytical techniques for the nitrosamine impurities. Majorly, we will go with the two techniques, either by LCMS or by GCMS. When we will choose GCMS and when we will choose the LCMS. If the molecule is volatile or semi-volatile, we will choose the GCMS technique. If the molecule is non-volatile, at that time we'll choose the LCMS technique. These are some major differences and advantages of LCMS and GCMS. Come to the regulatory overview for the current regulatory guidelines. Uh, Regulatory bodies like FDA and EMA have implemented stringent guidelines to control the nitrosamine impurities in API as well as drug products. Uh, then come to the permissible limit established as per the recent guidelines published by the US FDA in the year of September 2024. We can find it out the AI limit. Based on that, we can fix the specification limit for new NDSRI based on AA and maximum daily dosage. And also nowadays regulatory mandatory requires that pharmaceutical companies should require to conduct the compensative risk assessment to identify the impurities and also mitigate the nitrosamine contamination in the drug API and as well as drug product. Next, let's move to case studies. In our regular analysis, we will face the common challenges during the nitrosamine impurities. Out of that, one is presence of interfering compounds in the sample. That will leads to the complicates and accurate the quantification of nitrosamines. Okay, for this we have some recommended troubleshooting. Optimization of chromatographic separation. That means by adjusting the chromatographic conditions like mobile phase composition, column temperature, by using these kind of chromatographic separations, by using these kind of changes, we can increase the our resolution, or else we can avoid the interference between the unwanted peaks. Also, by employing some selective detection, utilization of particular modes like SIM or MRM, which mode we are choosing, that is very important to avoid the interference between other peaks. And sample cleaning. Before going to analysis, clean up the sample is the important thing to avoid interferences. Verify the peak. Whatever the peak eluded, which one we have to identify by injecting the standards. And identification of isobaric peaks. Whether sometimes what happens due to the isobaric peaks, we are going to get the interference. That one, if the isobaric peak is there, that one we need to confirm by HRMS or like Arbitraf and using other instruments. Let me move to case study two. Uh, in some times, what happens, we won't get the concurrent result between the two lots of a same batch. At that time, what we need to take the precautions, we have to examine the packaging condition. Means during the transit, how was the sample and what was the sample storage condition, but how during transit, how it was. These things to be taken to the mind for avoiding the uh, avoiding the result between two lots. Also, examine the environmental factors like temperature, humidity, and other things. Okay, and examine the physical nature. So, by visual observation, we have to check the sample condition, whether it is solid or liquid, and finally, we have to examine the solubility. If you have the COA of that particular lots, we can examine the solubility. Means after dissolving, whether both the lots are have the similar color and similar solutions or not. Based on that also, we can get the concurrent result between the two lots of a similar batch. Next. Let me move to my case study three, reference standard and quality of the reference standard. In many of our analysis, we will invent the or we will procure the standards from third-party vendors. But after getting the sample, we will inject into our instrument, and we came to know that the we won't get due to the bad quality or wrong standards received, which will result in the wrong results. So for that, some of the precautions we have to keep in our mind that we have to use the impurity certified impurity standards. That is the important thing, and we have to soon after receiving the impurity, we have to qualify it as our in-house impurity by testing at least minimum tests like mass, NMR. Based on that, we can identify, oh, whatever the impurity we have got is the correct one. This also helps to get the quality impurities. And qualifying the we have to qualify the impurity supplier vendors before buying the impurities. That is the good thing to get quality impurities. And consider the having solvents used means based on the COA we can check the solubility of impurities. Come to the case study 4. Issue was that it's a common issue. Sometimes we'll have a method in our hand or we'll receive some methods from customer end. Based on that, we'll start the analyze. But what happens? Detecting the nitrosamine exhibits the whatever the nitrosamine exhibited, that will gives the low sensitivity, which will leads to the suboptimal detection limit. So for that, some of the troubleshooting recommendations are first we need to review the sample preparation, whether sample preparation was done accurately or not, and extracted the maximum nitrosamine recovery or not. These things we need to priorly first soon after the uh, issue we have to check this one. Next, point number 2, we have to optimize the. Sorry. Point number 2, we have to optimize the chromatographic conditions. Means adjust the based on the changing the mobile phase composition, flow rate, and column temperature. We can increase the sensitivity of the our particular peak. Next, enhance the ionization efficiency by employing the different techniques like ESI technique. If you won't get the sensitivity in ESI technique, then we have to shift to APCA technique. Then by changing these kind of different ionization techniques, we can get the sensitivity. And calibration of instrument, this is also one of the major thing we need to keep it in mind, because keep on loading the sample. After that, after loading some sample, we should be clean our mass detector, and time to time calibration are important to get the good sensitivity from the mass system. Next. Let me move towards the case study 5. Addressing the matrix means sample matrix effect. It's a common challenges we are facing during our nitrosamine testing, that it leads to the sample matrix effect, leads to the recovery issue, means either poor recovery or more recovery. That's because of the presence of complex matrix complex in the sample, which causes the ion suppression or enhancement during the analysis of nitrosamine. For this, we have some recommended troubleshooting techniques. We have to utilize the matrix matched calibration standards. That means we have to prepare the calibration standard in the matrix similar to the sample to account for a matrix effect. That will reduces the matrix effect. Next, evaluate the various sample preparation. Means by using the extraction method. Whenever we are not getting the recovery due to the matrix effect, we have to change our sample preparation technique. So by using the extraction method, either maybe a solid phase or by a liquid extraction, which will minimize the matrix effect and improves your recovery level. And diluting the sample can also sometimes reduce the matrix effect and improves the accuracy or recovery in your analysis. And incorporating some internal standards, using the internal standards that are not affected by the matrix, that should be important. That whatever the internal standard we are using, that should not be affected by matrix. Then in that case also we can avoid the matrix interference, which will result in the improvement of a recovery. Mitigation strategies in API process. This is completely belongs to manufacturing unit. Means soon after analyzing the risk assessment, we need to mitigate the nitrosamine impurities in your process. If you assume if you have on 6 days reaction, in 6 days, where is the chances for forming the nitrosamine? Based on that, at that stage, we have to optimize the reaction condition by adjusting the temperature, pressure that can significantly enhance the safety and efficacy. And next, we have to select the appropriate raw material where the nitrosamine forming chances will be there. That will also mitigate our nitrosamine formation. Avoiding the nitrosamine agents. Avoiding nitrating agents. Excluding these agents via controlling the nitrosamine level in API also mitigates the formation of nitrosamines. And lastly, by using the alternate bases, because if you use either primary amine or secondary amine, if we can use the other bases instead of amines, there will be less chances to form the nitrosamine impurities that may be results in the mitigating the nitrosamines. Now conclusion and call to the action. Challenges and opportunities. Nitrosamine in pharmaceutical highlights the significant challenges but also present opportunities for innovation. Innovator support. Innovators are encouraged to support firms that optimize robust analytical strategies for better safety. Lastly, safer pharmaceuticals. Backing companies focusing on analytical strategies leads to the safer drug and secure investment returns. Um, it, with this, I'm going to conclude my presentation. Now, I would like to hand over to Dr. Nagraj. Dr. Nagraj, floor is yours.
Yeah, good afternoon once again to everyone. And first of all, thank you so much for giving your valuable time to listen to all these webinar session. So we are in the conclusion program. So this is the not end but the least, the Q&A session. So I am again requesting you while going forward, if you have any questions, kindly put into the Q&A session or you can chat with us. And today morning while registration doing for this webinar, we got some questions. I think some attendees are asking for that. It's a better way we can discuss with our colleagues. So the first question is asked by the one attendee. Are simulation studies better than the extractable studies for predicting the leachables? I think Suman, you should answer this.
Yes, this is a really good question because some of the clients are asking always whether we should do for simulation study or extractable study. So I'll give a general idea for what is simulation study. In a presentation also I have mentioned, but simulation studies mainly involve filling of a container closure system with a synthetic solvent or the drug formulation itself or the placebo, and exposing that one for accelerated aging condition to speed up the migration of the extractables. In some cases, we can say the simulation studies are better than the extractable studies. Here, the reasons are there that it provides a more realistic predictions of the leachables and their amount than the extractable studies. In the extractable studies, we are using a lot of solvents and different techniques, exhaustive techniques, so we can get a huge lot of extractables that might not leach into the drug product in actual cases. So simulation study is more realistic approach in case of that. And also it is also better than for leachable testing because it provides a valuable information to ensure patient safety without going for the full leachable testing like what generally we are doing. And also in while going for the leachable testing, we might face some challenges. Whereas in simulation study, because we initially from initiation we are using the placebo and the formulation for the external study also, so that analytical technical challenges will be easier to resolve and all those things. So yes, for some cases simulation studies we can tell is better than the extractable studies.
Thank you. Thank you, Suman. Dana, next question, please, on the screen. Yeah. What steps should manufacturer take to evaluate this risk of the nitrosamine? I think Dr. Kiran will be the best person to answer this question.
Yeah, Nagaraj, thank you. There are some steps as per the regulatory guidelines. First thing is that first we need to do the risk assessment for a particular drug or API. That is the first step. Next step, confirmation test for the confirmation test after the risk assessment. After that, third stage is reporting those confirmed tests. Next, that mitigation, impurity mitigation study. Based upon that, we have to update the marketing authorities. Based on that, we have to submit the report to regulatory authority.
Okay. Thank you, Dr. Kiran. The third question will be why extractable and leachable study is mandatory by the European Medicines Agency, followed by FDA. Suman?
Uh, External leachable study has become mandatory now for any NDA submissions for any drug product. It is for all the regulatory for FDA and EMA and other regulatories also like ANVISA. Because it is crucial for ensuring patient safety by identifying and qualifying the potential chemicals or contaminants that can migrate from the packaging materials or the manufacturing equipment into the pharmaceutical products. And potentially, it will impact the quality, the efficacy, and also the safety due to its interaction with the drug substances. It's mainly the studies essentially helps in guaranteeing that drug products are not contaminated by chemicals leaching from the containers or any production system. And yes, that's why it has become mandatory for all the regulatories uh, to do this study for submission of for any NDA submissions.
Okay. Okay. Fourth question, please. Yeah, what actions to be taken in case of any unknowns are observed above the AET while performing the extractable leachable study? I think Suman, you will answer this
Uh,
question.
yes, also yes, is most of the cases uh, it is fine if we get an leachable above 80. Yeah, the first things that we need to do have some more experimental that study will prolong for that. So additional experimentations is required for the identification and confirmation of that particular leachable, and toxicological evaluation is required mainly with help of structure activity relationship and all those things, and we need to assess whether that leachable is carcinogenic or mutagenic or genotoxic. So we have to assess for those. But I can say that if anything is coming above 80 level, so yes, that we have to do. We have to identify if standards are known or available. We have to quantify that. We have to develop a method and validate the method for that one. If standards are not available, then with the known standard, we can qualitatively quantify those leachables which is coming up 80 and validate those method, and then uh, we can check for the stability batches for defined lifetime up to the shelf life of that product.
Okay. Thank you, Suman. Dana, next question, please, if you have any. Acha, how fix? Okay, this is a good question. So how to fix the specification limit for new NDSIR? Dr. Kiran?
Nagaraj, thank you. It's a really good question because this is the major thing nowadays fixing the nitrosamine impurity specification for nitrosamine impurities. That too for nowadays NDRS era is going on. For that, we can fix the based on the acceptance intake divided by maximum daily dosage. Calculation of acceptance intake for some APIs already regulatory has published the AI value, or else we have to calculate the AI value by using the FDA guidelines published recently in the year of 2024, September 2024. There are some steps. Based on those steps, we can calculate the AI value for our moiety. If we got the AI value, and it's very simple to calculate AI divided by maximum daily dosage. We can fix your specification limit for any new NDSRIs.
Okay, thank you, Kiran. Acha, as discussed earlier, I got one question in Q&A session. I think attendee's name is Anishta Rai. She would like to ask that we were undergoing a lamotrigine investigation for black spot in finished good during stability studies at accelerated long-term and ambient conditions. So nitrosamine study was done for API and other excipients also. Looking at what your study for rubber curing and other packaging material, could the black spot be a result of nitrosamine of a packaging material? Is is just a possibility or how?
So
Suman?
for that, we need to check for the rubber stoppers and all those things. Like I discussed earlier, that we have to check the rubber stoppers for any secondary or tertiary amines which can convert to secondary amines, and then we can conclude that any secondary amine is present in your rubber elastomeric materials or not. And if anything is present, there might be a possibility for that your black spot is coming, or for any other chemical reactions also with your drug product, that it might react and it might get. But initially you have to check that also. And also because of your migration of ink from your labels, that also might result in black spots. So we have to do a thorough extractable studies for your rubber stoppers and also your printed labels and cartons or any stickers, whatever you are using, to get an idea from where these black spots are coming.
Okay. So also regarding this, she would like to ask in detail, what is the general average time of getting the above acceptable limits of nitrosamine for general packaging material like rubber and plastics? So this question comes with the food containers, like that she told. So what will be your suggestion on this?
See, depending upon your concerns, that for thorough extractable study and this investigation, uh, it is requires some time. Means depending upon what are the regulatories and what they are asking, because different regulatories are different, and things in in India, FSA is still working for other things are there. So we have to check on nitrosamines and all those things. We have to check that average time or getting acceptable limits are still not defined for these. For outer countries, they have defined some limits for those, some acceptable limits, but India still they have prescribed some limit, but it is not so much stringent over here. What time it will come or not, that is very difficult to say for me right now. But hopefully
Okay.
in the near future, because now all these problems we are facing with nitrosamines and all those things, I think very soon we'll get stringent limits for those also. And the main case for that thing also, the source for the production of nitrosamines and all those things is very different for the food and other pharma packaging materials. So yes, it might take some time. We can surely get in near future that limits.
Okay, okay. Thank you, Suman. So, behalf of Shiva team, I would like to say thank you for asking this question to Rai Madam, and I am requesting you. I am going to share in next slide our contact details, where you can directly contact with us in detail about these questions. If possible, if you are not satisfied with the answer, we can arrange a Teams call, because we are always looking customer as a long-term association, so where they can satisfy their expertise and questions by asking to get a confidence on us. So I think in Q&A session only I got one question and followed by earlier five questions. So six questions answered. My team, the analytical team is given the answer up to the mark. And so these are the contact details. So if you have any doubts, clarity, kindly contact us. We are happy to assist you 24 by 7. And while going forward, I think Again, I am going to say all big thank you to all webinar attendees and special thanks to Satya, Dr. Kiran, Mr. Suman for arranging all these. I think one more question I'm getting from just a minute. Okay. So thank you once again to all of you. And I'm taking conclusion part that hope this webinar session had given you the best knowledge about the Shiva's capabilities, especially on the API and finished products in terms of nitrosamine and E&L studies. And hope this webinar is beneficial for all of you and looking forward to get your great question where we can prove ourselves by giving satisfactory answers. So thank you so much once again to all of you.